I guess we wanted to check in, kind of how things are going, your analysis and things. So I'll let you give me a quick summary of what, if you have any questions or what things that you've done so far.
I'm doing good. I think I'm done with 4, and I have remaining 3 with the everything what you have told me, because it took time to
Right.
process. I was like seeing everything, like, and
Yeah.
then it is good. I understood everything. I'm getting into track what to do, what not to do. All this. This is interesting.
Great.
This is interesting.
Yeah.
So
Yeah.
we have like to complete this probably in today by evening 4 I can complete this.
Oh, okay.
Yeah,
Great.
so this will be as you taught me on
Mm-hmm.
last week you taught me that will be done by today evening for all of the
Wonderful.
files, including the
Yeah.
cohort analysis.
Oh, okay. Yeah, I was going to ask about that, how
Yeah.
that's going.
So, but now you said you will teach me IGV and that. It is your wish. You can
Yes.
teach me tomorrow or you can teach me today. So, yeah.
Either one. Yeah, we can
Look,
kind of
depending
start
on your
getting into.
schedule and all that.
Yeah.
Uh,
Yeah, we can get started on it today. And then, it's one, if you have any variants that you're interested in checking, for like their quality and things. So I'll do like just an overview of IGV today. And then if you have, like, once you finish your analysis of things,
okay.
If there's any variants that you feel like that you think are super interesting and you want to check their quality in IGVs, because that's kind of like one of the last steps in this whole process to making sure, like, I really like these variants, and now I need to make sure that, they actually look good quality and are worth pursuing potentially for some other things. So when you finish your analysis, if there's any handful that you would want to go check, we can do that another time.
Okay.
Later this week, we can do that. So unfortunately, it's getting access to the raw data files which we use for IGV is a little more complicated. So because they're located on our server, and just getting people access to that is a little bit of work. So that's why I think if you have any specific ones you want to look at, we can always do that together, and I can pull up those files for you, because it's not impossible for me to do it, like just to say, hey, go look at these files on your own, unfortunately. So let me go ahead and start sharing here, and then we'll do some. First things first is connecting to the server. Do that. And I think we'll just do an example case right now, because I actually would have to see where the cram files for your files are. And I think I may need to ask Hannah about that. Because I know they came from an outside place. So they may be somewhere else. Okay. So all of the files are... Actually, I think I need to go to here. All right. So a lot of the samples that we've processed from a lot of our old cases and things were run at the core facility that Hopkins has. It's one of the other buildings on campus. It's their division is called CIDR, Center for Inherited Disease Research. So they run a lot of samples for not just Hopkins people, but anyone worldwide that wants to utilize their core services for sequencing. So that's I have everything here in the CIDR folder, and then the CRAM file folder is where most of them are located. Because a CRAM file is basically a condensed version of the BAM file. So I don't know if we've looked at this table, this kind of graphic together. Let me find that
where
for you.
There is codes, like the binary file where there are a lot of codes.
Yeah, so that the BAM stands for binary alignment map.
Okay.
And that's what the computer uses to read all of the data. And then the CRAM version is a condensed version of that. So just like you would zip up a file for like emailing or something, and then someone can expand it when they receive it. It's a similar concept to that. So they would do, I just wanted to just kind of point this out. You may have looked at this table already, but I just wanted to show it to you because I think it's a nice visual example. There we go. This one.
Oh.
Have you looked at, I don't know if you looked through these slides before, but this is one of the teaching slide decks we use for when we're teaching people about more like the wet bench part of the sequencing process, or when the samples are processed. So once all that wet bench stuff is done, and it comes time to actually put it through the scanner. Typically, they're all run on this NovaSeq 6000 scanner right now. And what the scanner produces are something called FASTQ files. So FASTQ files are just a whole pile of these short reads. And the reads are, for short read sequencing, they're about 100 to 120 base pairs in size. Longer reads are the reads are KB in size. They're, like a long read sequencing, be very large pieces. But for short reads, this FASTQ file is, all the little reads plus a quality score of how well that read performed when it was scanned. And so now that you have a pile of reads, you then have to go through an alignment process to be able to align all those reads back to a reference genome to get them in the right order of the sequence. So you utilize an algorithm called an aligner algorithm, which two of them that we have used before, BWA or Bowtie, are the names of those algorithms. And once they are now aligned, all of these little reads are aligned to the reference genome. It then is called an alignment map. So it's either called a SAM, which is a sample alignment map, or a BAM. The SAM is something that's like human readable, and the BAM is the binary version of that file. And so now that you have everything aligned back to the reference, you then have to identify where the mutations are in your patient sample versus and compared to the reference. So every time that something is, like this red spot right here would be like a position of a mutation in the patient. And you can see that it's present on, multiple reads of that same base position. And so you would now have to run something called a variant caller algorithm. So haplotype caller or MUTEC2 are two that we've used in the past. So then once it goes through and identifies all these places where the patient is different than the reference, it creates a file of all of those differences, and it's called a variant call file, the VCF file. So it's just a list of the sites and what type of mutation it is and like what that genotype is. So that's the standard process. So the VCF file is what we actually put into PhenoDB for the analysis. We're just looking at all those list of those mutations. And then we annotate them for all of those columns that we utilize.
Okay. So
back in the binary alignment map stage, this is what we're going to be looking at in IGV, is this view right here. So it's before, and it will show you the mutations and where they're different than the reference, but it's not anything that's actually like you're not going to be having just a list of the variants itself, like the VCF file. So that's visually, I wanted to show you this just to get a perspective of where we are in the process of what data we're looking at.
And the one, like if you go to the back, the Nova sequence 6000, the Illumina, that is where we keep our DNA.
So here, I'll back up just a little bit. This is the best place to put this. Okay, so all the chemistry of the actual assay itself happens on something called a flow cell, and so the DNA you have a mixture of DNA and all of these little adapter primers and things. So you make the little mixture of that. And you add it to these lanes of this flow cell.
With the
And then,
pipette like this small.
yeah, they pipette it into these little wells down here at the bottom. And then by like a capillary action, it sends that liquid all the way down the lane of the flow cell. And then, so on the lawn of this flow cell are all these little, let me see if I can make it bigger here. Let me do this.
Then we get a print through the machine.
Yeah, so what it does, so you have these adapters and your DNA attaches to these adapters, and an enzyme goes through and actually adds the next base pair sequence. So this is, the first, out of the 100 base pair read, this is read, spot number 1, 2, 3, 4, 5, and so on, because the enzyme will come in and add the complementary base to these adapters. So there's a lot more that goes on to that, but that's it in a nutshell. So once all of these sequences have been added by the enzyme, you then do all of these nucleotides that have been added have a fluorochrome attached to them of a specific color. And then, so when you put it on the scanner, the NovaSeq 6000, it has a light that will shine and then basically fluoresce these different colors. And it will then, if you go back here, it's a little hard to see. Like, this is a little visual of the everything fluorescing as it's been illuminated on the scanner. And then the scanner will take a picture of that, and it will know just to add each, for all of these thousands upon thousands of little spots, it will keep track of what, each picture and each one that it illuminates. So it does multiple rounds of that. So how many times this, a particular locus's base pair has been sequenced, that's your depth of sequencing. So so then everything's illuminated and you get the colors on the scanner then so you have and it reads it for like 100 or so base pairs for each of those reads. Mm-hmm. And so that is then what is taken in that file that you get at the end that's your FASTQ file. So all these little short reads and what their like color sequence is, which is then translated because each base pair, each A, T, C, and G has its own color, and then that is translated into the nucleotide code. So does that make sense about the process of that?
Yes, it does.
Yeah. If you ever want to know more about like the actual nitty-gritty of what happens on the flow cell and everything, if you there's a really good video on YouTube. If you do Illumina sequencing by synthesis, then you can watch that, and I like that video because it just explains things very nicely with little pictures and cartoons of what's happening. And so I like, if you want to go see that, that's super interesting. So we're going to be looking at the state of things in IGV.
Okay.
So let me get out of this. Go. And IGV, Integrated Genome Viewer, is what IGV stands for. And if you just Google that, it's a product of the Broad Institute, and you can download a free version of this. What we're looking at here. Mm-hmm. And then you can bring in your own files from your local machine or your server or something to look at it. So everything that, the first thing when I first open the program is I want to make sure I have the right reference sequence that will be displayed here at the bottom. Mm-hmm. All my patient data is going to be up here, but my reference sequence is going to be displayed at the bottom. So our sequence that we use is the build 37 sequence. There are other versions, that are more recent, like build 38 and et cetera. But we use build 37. So that's the reference that I have loaded in. Mm-hmm. So from here, let me just see which file we want to bring in now. Let's find something potentially interesting. I'm just going to find a file that I did a long time ago. And there we go. Let's try. I'm gonna open the file. There we go. Not a lot to look at in this one. These are all. I want to get some with a lot of good different examples in it. I don't think this one's up.
Mm-hmm.
Yeah, not all of these do I do IGV for. So that's kind of what I'm searching for right now. Let's try this. Okay. This is kind of a good, I think, overview example here. Okay. So let's do 15291. So every time, while this is thinking, the file name or number, so every file is tagged with a number, and it comes from what the scanner tags it as. Mm-hmm. It's called an SM tag, mm-hmm. And so I have to look it up by that number, which is what's present in GenoDB here. Of course, it's going to take forever. Log on.
You can once cut the tab and go to the new tab, and then you can mendeliangenomics.org.
Oh, what is that? Do a new tab?
Yeah, and close the last tab.
Oh, the other one?
Yeah. And here I think it's better when we go to, I think it will
open.
Go. Yeah.
There we go. 15291. Okay. So this is a case that has the proban, mom, and dad. So it's a nice standard trio. Mm-hmm. And so these are the numbers here that I'm going to be focusing on. This is my SM tag number. Okay. And so now I need to go back to IGV. Real quick, I'm going to move this to my other screen so I can see both numbers at the same time. All right, so I'm going to load from files. Mm-hmm. So these files are very large. The the whole exome files are, a few megabases in size. The whole genome files of BAM files can be like gigabases in size. So it's a lot of data to open with. So it might take a couple minutes for it to actually load this whole file, because it's just a whole huge file of BAM files of all, hundreds of cases. Mm. So it will take us a moment, but this is kind of the slowest part of this whole process, is just getting everything loaded in, honestly. So it won't be too bad eventually when it opens. So yeah, I'm hoping I think this file will have some example of what like an insertion or indels will look like, and then what a single nucleotide type variant will look like as well. So we'll get a sense of all those things. So this, I'm doing 13906. Go down a bit. 139. Oh, too far. There we go. So you can see that for each file name, there's 3 different files with different extensions on them. There's crai.cram.cram.crai. So the crais are like index files that are, they're also included when you run the scanner and things. So I don't need to actually select those.
Okay.
When I bring in, I just need the .cram file itself. So I can just hit shift and then I can select all 3. And so here's patient, mom, and dad. So those are the 3 that I want to bring in. I'll hit open. And it'll take a minute or so to load that. And then once that's done, then we'll look at each of our variants in this file that we have here. There we go. So when you first bring a file in, you're not actually localized to any region yet, so it just kind of starts up like this. So the first one we'll look at is, let's do a nice pretty one first. So we'll start here on chromosome 1. So I can highlight from my Excel file my chromosome and my start position together, and I'll do a copy. And then I can just switch back over to IGV, and then the search bar at the top, paste that into there and hit enter, and it should proceed to that spot. So the first time I open up cases and bring them into IGV, I have to just tweak something in how the data is visualized. So everything's in gray right now. So I want to change the coloring of all of these horizontal reads that I have here. So I can just right click on one of the reads and do color alignments by read strand. Okay, and I'll just do that real quick for all 3. Oh, no, I took that. Okay. So now everything's looking good. Let me just zoom out for a second to show you. So you can see that, so this is a 120-ish base pair read. And so when the sequencing is done, it's bidirectional. So one read is sequenced in the forward direction, and the other read is sequenced in the reverse direction. So the forward reads are colored in red, and then the reverse ones, you can see that there's an arrow at this end for the forward primer and an arrow at this end for the reverse primer on the blue one. So blue is reverse and red is forward. So when I want to be able to see, and I can scroll through, and you can see that there's a very large depth of coverage because all these reads are stacked up with one another. Let's zoom out a little bit more. And you can see how they all are aligning back to a reference sequence. And my reference sequence, zoom back in, is down here at the bottom. I can see that all the sequence of letters down there. So when I copied and pasted my first site I was interested in chromosome 1 here, it zooms it right to the middle of the view. And so you can see that in my Excel file, the reference is a T and my patient's been mutated to a G. So at this base position, you can see the reference is a T, and if I follow it up, then I can see there are reads that I have a G, G, and so forth.
Why did you look at
the
exact Q11 position?
This one, or like we're at this base position here, 9272.
So that one is okay. There, the 3-2. Oh. Okay.
Yeah. So if I click on, it should go right to the middle of my cut and pasted my thing into the search bar, right?
Okay.
So it's in this position right here in the middle. And if you click on these little bars at the top, these gray bars at the top, I can actually see that this is my base position, chromosome 1, 32,829,272, which is the same base position that I have from my Excel file here. So that's how I know I'm looking at the same spot.
Okay.
And you can see that there's a total read count of 133. So that means this base position has been sequenced 133 times from all of these reads. So if I scroll down and I would count literally every single one of these horizontal lines, that would add up to 133. Oh. So anytime that there's no letter there, that means it's the wild type, which is the T. So it's not going to display it. But if I have a mutation there to something else, it will display it on that read. So this read is showing a G. All of these are wild type. This one's a G as well. And then I can just see the distribution of wild types to mutations as I scroll down that spot. You can see that this read has like a T there instead. Because this read is showing all of these different things, I would classify this as like a poor read. This kind of they happen erroneously. This is why you have the depth of sequencing there, because all these other reads look very pretty and very nice.
Yeah.
So when I click on this little bar at the top, I can see that 84% of the 133 are showing the wild type, and then 16% or 21 reads are showing the G. So that's good. When I do that, I add a column to the beginning of my Excel file for IGV comments, and I can just say, yes, this one looks real. All my reads look like they're good quality. I'm seeing a distribution of wild type to mutant alleles. And I can, if you click on the actual read itself, it will tell you even more in-depth information about like the mapping quality score that this one has, and things like that. So the mapping quality of 60 is the highest you can get. So this is a good looking one. And you can actually also tell, because sometimes when you get poorer quality reads, instead of like red and blue, it will then display the color as like a very light pink and then a light blue. So that's how I can kind of get a quick sense of like, oh, this read is a little bit poorer quality as well. So it's nice that it does that. So I'm seeing this in my patient who's listed here at the top. My mom is in the middle and my dad's data is at the bottom. So I do kind of run myself through mom's data, because this is an autosomal dominant new mutation analysis. So I shouldn't see, the reads in either of the parents, and I should only see the mutation in the patient because it's supposed to be a new mutation. So that's good. I don't see anything in mom, and then I'm also not seeing anything in dad. So that's great. And the reason I definitely want to check the parents is because sometimes the variant caller, it's pretty darn good. But every once in a while, let's say there's fewer reads seen in maybe one of the parents. So because it didn't reach that threshold of being able to be significant enough to call it, and then it wouldn't have been, then it's not going to make it to your VCF file. And it's going to make it look like this is really de novo in the patient, when really you are actually seeing reads as being, having these G's present in one of the parents. And if that's the case, I have to do a little bit more digging or homework to be like, to kind of figure out why. Maybe the depth of sequencing in the mom or the dad was just very low, and you may only have, out of 10 reads, you only have one that's showing up as a G. So in that respect, then I would be suspicious. Maybe my depth of sequencing just wasn't enough for my parent. So I can't get that enough, it's not detecting that G enough times to, actually be being called. So in that case, I would then want to run a validation assay, like a Sanger sequence, to see if with all three, mom, dad, and child, to verify that, there really are more reads in the mom, and she really does have that G. So therefore, it's inherited from the mom, and it's no longer a de novo call. So you kind of have to think about that. But in this case, I don't see any reads at all that make me suspicious of and the depth is beautiful for both parents. You see there's a whole ton of reads. There's a depth of 81 for the mom, and then a depth of 131 for the dad. So both of them have a decent amount of depth. I don't, when I get like less than 20, I get a little bit more like, is that really enough? But it's not so bad. These counts are great. So this one's beautiful. This one's nice.
Yeah.
So the next one then I would just keep working my way down my list, checking all of those future things. So this one, let's go to this one next. This is a chromosome X, and this is how a single nucleotide variant would appear. You'll have your reads with your mutations, your different mutation letters showing up on those reads. So for an indel, for an insertion or deletion, this one's an example of one base pair deletion. It's G in the wild type, and it's not present in the patient. So let's go look at that one. So I'll just copy that, and I'll paste it up here in my search bar. And it's going to, when we switch between chromosomes, it takes a minute to reload all the data. So this is the second most time-consuming part of this process.
Okay.
I find that if there's a lot of, it's faster when it moves within the same chromosome. So if I have a whole laundry list of variants to be checking, then I will maybe bounce around. And if I have chromosome 1 up here and here and here, I will check all the ones first for speed, and then I'll go back and check the others. So we've loaded the track now. So you can see that this area has much less depth of sequencing than that first one we looked at. But just visually, you can see that there is now a gap in this read. It has the little black bar. So this means that this one nucleotide is not present in this sequence. And it's supposed to be a G on my reference, and that is what I was seeing in my Excel file. And then the patient has a few reads that are missing. So clicking on this bar at the top, there's a total count of 8 for my depth of sequence, and I'm seeing the deletion in 2 of those reads. So that's fine. It is a lower depth area, and you can see that the mom's only has a depth of 7, and the dad has a depth of 5. So he's even less. So none of them with those numbers are showing any reads that have a deletion on them. But I now wonder, is it just because there wasn't enough? Do I need another 3 reads that for either of these parents to be able to actually see the deletions show up? I don't know. So therefore, it's looking real in my patient, but I have in my notes I wrote low depth in parents. So I'm going to question that I'm not 100% sure right now that this is really a de novo call in the child, because I didn't have enough sequencing depth in my parents to really be able to tell that. So in this case, as you've done all your prioritization and everything and whatnot, if you're at this point, I would then, let's say this particular call in this gene has a lot of really good things going for it based on your prioritization. You really like it. Then I would take the time to say, yes, let's run a Sanger sequence to validate it, to really see if it's inherited or not. Okay. Because, that does take a little bit of work and time. We don't have enough time and resources to literally Sanger sequence all 17 of these variants, right? That's a lot of work. So I go and make my notes about everything. Then I look and see how many of these were super interesting to me, to then say, hey, this one, I was questioning this one, but I really like it for a lot of other reasons. I'm going to go ahead and Sanger it to confirm. So that's the logic behind it. Let's look at this one real quick. This is another deletion.
So we are just confirming the start position. Means we take the start position. We chromosome and the start position. We keep it in there. We check the reference gene and the alternative gene.
Yep, just to make sure I'm really looking at the right variant.
Further we are verifying what is given in the file and reconfirming it is correct or not. That's what we are doing right now.
We're confirming the quality of the reads. How good are the and I think
based on the dark or light color, we are telling how good are the reads.
I'll show you some examples of that. Some of these ones up here where I've made a mix of good and poor quality reads.
Because until now I have been looking at the good read, so I don't know what is the poor read.
Yeah, I'm showing you the pretty stuff first. And so now, this is a chromosome 9, 3 base pair deletion of a G, G, and C.
Okay.
So this is my GGC, my reference. and I see that now this, because it's more than one nucleotide now that's being deleted, it's 3, so I have this number 3 appear in the middle of that black bar.
Okay.
So I can see that it's a decent, a rather decent depth. What is this one? This depth is 53, and I'm seeing a deletion in 2 reads.
Okay.
And now I'm also wanting to see the parents. They have a decent, more decent depth here, but I'm not seeing any deletion reads in them. So I do feel good that it's not due to a low depth of sequencing that I'm not seeing it in the parents. So I really do think this is probably de novo. That's fine. But one other thing, especially when you see non-frame shift variants, so either insertions or deletions like this one, I want to make sure that I'm also checking my reference sequence.
Okay.
So this is a GGC, then I have another GGC, and another GGC, and another one, and yeah. So I'm seeing it's much like the TRF aspect of things, these polymorphic repeats, trinucleotide repeat regions. This is a smaller one. I'm only having 1, 2, 3, 4, 5 repeats of this GGC.
Okay.
So whether or not this one that's happening in my patient here, it could just be that the enzyme got a little confused while it was going through its sequencing process, because in these repetitive regions, that's where it gets confused the most. It has most trouble with those regions. So in my note, I did say, quality-wise with my reads and stuff, it looks real to me. But I did make a note that it's in a small GGC repeat region. So, and also because it's non-frame shift, I'm probably not prioritizing it very high when I go through my whole process and things. But it's still nice for me to see and to have confirmed that this is a kind of a repetitive region even to begin with.
Okay.
So that's why I make that kind of note.
Okay.
So this one, let's just, before we jump up to those ones with the poor quality, I want to go to this one on chromosome 18 here. Because this is one where I think I've seen a read in one of them, in the mom, that may or may not be significant. So we'll do that. Okay, so this one, and see, this is the little gray bars at the top. This time you have one that is, it's color coded now, because when you get to, I'll click on it here. So this is a nice, this is supposed to be a heterozygous call. So you want 50% of your reads showing the wild type and the other 50% showing the mutation. And that's an approximate number. So this is 45-55. So this is a pretty darn good, nice half and half split between mutation and wild type. Some of the previous ones we saw, I think it was 18% and then 80-something% of wild type, that first one we looked at. That's still fine. When I get beyond that 80-20, so let's say it was 10% mutation, 90% wild type, then I get a little suspicious that maybe I'm starting to, is it actually a false positive call? Or am I potentially seeing mosaicism in my samples? So that, mosaicism is when just a small portion of your cells have a mutation and all the rest are wild type. It's not seen in every single cell germline that you might have. Yes. So to be able to confirm mosaicism, I would then also want to run a Sanger, because that depth of sequencing is a lot higher, and you could potentially start seeing the difference in the percentages a little bit more clearly. And so, but you start to get the suspicion, maybe mosaicism, if you're outside of this 80-20 range of wild type to mutation. But this one is a beautiful half and half. So here you actually get the color codes of the red is for the T and the blue is for this wild type C. So you have the half red, half blue on this little histogram thing. So here I have, what's the med depth again? So out of 42 reads, 23 wild type 19 mutation. In my mom, I can see, look, I see one little T there. She's only got 12 reads, though, and one of them is showing the T. So this is where I would be a little suspicious that mom, I see, so I wrote my note, low depth in parents, 1 read in mother, 1 out of 12 reads.
Oh.
So again, if I'm really liking this variant, I would need to Sanger it to see if I really like it. So really, this is where you got to play to both aspects of things. We check them all, and then what I decide to discard is really things that are terrible quality, the things that I really don't look like a false positive. And then you have to look at things that are a little suspicious and compare them to your prioritization list. And things that are high priority but suspicious, then you have to do some confirmation testing before moving forward. So there's that. All of this, my homozygous mutations here are all in chromosome X. Because this is a male patient, all the X calls for him look homozygous because there's only 1 copy of your X. But when I actually go to that call, it's going to actually be showing homozygous in the mom, because obviously the male inherits their chromosome X from their mom and the Y from the dad. So the mom's going to be heterozygous for her. One of her chromosome X's has this call, and the patient obviously inherited that one. So all of his variants here, this is what a homozygous one would look like. 100% of all your reads are going to be showing this A. You can see how all of them have it, versus like a heterozygous call where it's about 50-50. You see the difference?
Yes.
Yeah. So that's why in my notes here, I just wrote, it looks real, but it's heterozygous in the mom, which is to be expected because, it's an X. So, all right. So let's look at this weird 14.
Okay.
See what's going on here.
So I think IGV is interesting. We need to go through the variants and we have to further check.
Yep.
And then comment.
Exactly. So a lot of times, if I have a huge list of variants, I will definitely be checking all of my indel calls versus my SNV calls, because the quality of the data has gotten very good for calling single nucleotide variants. Where the enzyme sometimes has the trouble is with the indel calls, possibly because of repetitive regions, but you never know. So I definitely make a point of checking all my indel calls to see what the quality of those are. But sometimes my SNVs, I will check too, especially ones that I'm calls that are very high on my priority list as well, just to verify that they're 100% look very nice. So this is supposedly a C to a T. Here's my C right here. And as I follow it up, I am seeing my T's. But you can see a lot of them, like I mentioned, the color is different now. Like a light pink on this read and a light blue on this read. So you can see that there are some good quality reads in there. But I'm not really seeing a lot of my T's appearing on those nice reads. A lot of them are appearing in those light colored reads. You see that? So this makes me suspicious that this call is actually real or not. Okay. Because, I have 2 that are appearing on my good quality read. I do have more as I go down. But in general, I'm seeing a lot of them appear on the poor quality reads. So this is going to make me say it's real. I'm also actually seeing it, Mom, we'll go back and look at that. And you have a mix of good and poor quality reads. So so I've scrolled through and seen all this on the patient, right? I'll scroll through again. Look, I'm seeing T's appearing in Mom as well. But the variant caller just didn't pick it up as much as it did for the patient. So it makes it look like it's de novo. Actually, this is the compound het file, so that makes sense. because I think if we go down to the parent column, here we go. The parent column is here with the M's and F's on it, for mom and dad. So I am, but I also like to note it in my IGV comments as well, that yes, seen in mother. We actually have 3 calls for POT, EG, and you have two that look like they're coming from the maternal and 1 from the dad, and which makes sense because I'm not seeing anything in the dad for this call. But I did make the same note. This region is obviously very messy because the read quality isn't great. We'll just bounce over to this one now. Yeah, so here's the chromosome A, the other one for the A mutation. I'm seeing it in, excuse me, why I'm opening that. So here, if you click on, and I click on the mapping quality, my mapping quality is 0 for this read. So it's cruddy. So that's what I would then say. So I'm really not, and it looks like this is the same thing for this other variant, chromosome 14 and the POTEM gene as well. So I'm not terribly excited about any of these reads, honestly. So I probably would prioritize, if I really, really liked these from all my prioritization information, I would be very surprised that I really don't, I'm probably going to move them down my list a little bit because they don't look very real to me. So that is, in a nutshell, how we go through and look at IGV and what we look for. I'll just mention real quick, let me go back to this one with the deletions on it, because I don't have an example of an insertion call. So if I had an insertion, it would actually just be, if it was a 1 base pair insertion, it would show up as a letter I, right here at this base position. And then if it's an insertion of more than 1 nucleotide, let's say 3 nucleotides, it would be an I with a 3 in the middle of it. That just tells you how many nucleotides are supposedly stuck in there.
And the gap will be there, just like this?
No, it's not going to be a gap. It's just going to be, I don't have any good, let me see if I can.
Just pink it will be, that's it?
That's actually purple, but yeah, it's a little purple thing for an insertion. Let me go to another. Actually, maybe this one is one. Here we go. So this is the I. That's the insertion.
Okay.
Yep. So supposedly there's a little insertion right at this base position here. And then if it was more than 1 nucleotide, it would have the number of that in the middle of this I. It would say 4 or 12 or however many base pairs are supposedly inserted at this spot right here. So that's how you're going to see the insertions. It's this little I guy. So, yep. Well, I think that's it for example-wise.
Yeah.
And a lot of times, just in general, while you're going through and looking at these, some of this stuff that you're looking at can be rather subjective for, of how I don't think this is a really great quality, or I don't think whatever. So if you ever have a question about it, so sometimes I'll tap one of my coworkers and hey, what do you think of this call? Do you think it's a really decent quality or not? It happened a lot more back in the early days of the sequencing that we were doing because the data wasn't as clean. The assays that they're using now have gotten so nice. So I don't find that happening as much anymore, but it can happen. I'm just kind of reminding you that it's this is a subjective process. You kind of have to, if you ever have a question about it, it's not unusual.
I will ask
you can definitely ask. So, yeah.
Okay.
Any questions in general about this?
I understood.
Great. Oh, I want to do just one quick little other thing. So you can see how you can also see that the gray bars of the histogram here are how tall they are or how far the depth of sequencing is. So this one, so you can see that, let me move this down. So this is this is gene HABP4, right? So within this gene, this is exon number 1.
Huh.
And if we scroll down to exon 2, you can see that there's another peak of sequencing over exon 2 because this is an exome data. So you don't have, we're not sequencing hardly anything in these non-coding intronic regions here. You can see there's a few stray reads, but not anything like major like here. So you can see that the peak happens over the exonic region. And like I did mention, we sequenced a few base pairs into the intronic region to capture the splicing areas. So that's why you have the bell kind of curve happening. And then you do also kind of wanted to keep in back in your mind that there are some regions, even if they're exonic, that are very hard to sequence. So you may get a depth that is very, very low for a part of an exon, or there may be an exon of a gene that has like 10 exons, but exon 1 is very hard to sequence, so you get hardly, if any, reads on exon 1.
An example?
Sort of a little, I think it might, if I zoom in here, let me see, this might be a good example on this one. This one's not down to 0, but you can just see how the depth, even within the middle of this exon here, the middle, like you have nice sequence, and then just drops down to hardly anything, like right here in this region of this exon here. And there are some times where that depth will drop down to 0, and you'll have no sequencing as part of that. So if you have a variant in a gene, might be, it's for a recessive condition where you have to have 2 variants. But let's say you found one variant in one part of the like exon 4, and that phenotype looks really good for your patient, it's matching really, really well, but you're not, the sequencing data isn't finding the second variant in that gene. It would be helpful to go through and look at your depth of sequencing to see if there's maybe an exon that's not well covered, and say, oh, maybe the second hit is hiding somewhere in exon 1 that doesn't have any coverage. So then you have to try other methods to try and sequence that region to maybe try and find your second variant. So, that can happen from time to time. So that's just another thing to kind of be aware of. If, you're finding one hit and it just matches so nicely but the mode of inheritance doesn't correlate because you only have one variant, maybe you're missing another variant because either it's in a non-well-covered exon, or maybe it's actually a non-coding variant that actually does have some function that we're also missing because of this exome data, and I'm not sequencing non-coding regions. So that's a lot of times, not a lot of times, that's sometimes what happens when we run a whole exome for a case and we don't find any good strong candidates. It might just mean that we need to maybe send it for whole genome sequencing to find something that way. So there's different pathways and avenues if you to try and see more about a case. So there's a case from our Mendel project days, a very old case that we sequenced. Literally any new technology that comes up, we started with that very first exome and then didn't find anything for. So then we held that sample for a while, and then when whole genome sequencing came out, we sent it for that. She's also been sent for RNA-seq as well. We sent her for long read whole genome sequencing as well. So we are still on the hunt for her answer, but some of these cases do happen that are, very elusive for finding those variants. So yeah, so that's IGV, and I think it's very helpful, and it's really important to look at the raw data too, and not just trust blindly what you see in your Excel file, because it is important to also double check things, I think, sometimes. So, but yeah, all right. Well, so once you finish going through your prioritization and your cohort stuff, we can check in about that, and then if there's any interesting variants that you want to look at for
So yeah,
IGV,
and
we
you
can
will
do
help
that
me
as well.
with that, like.
Yeah. I also should, I'll write myself a note to try and find out where your cram files are so that we can do that.
Okay.
Because that's going to be important as well.
Okay.
I'll have to go on the hunt for those today. Genochondromatosis cram files.
So you all store the cram files in a database?
So they're on a server, just on there on its own. We don't store them within PhenoDB because they're too large and they're very big, and it would just drag the speed of PhenoDB down very slowly. So that's why we have to store them on their own server. And that's also why we store cram files versus BAM files, because they're condensed and a little bit smaller than the big file itself. That's a big problem. A lot of labs had was just data storage and the cost of that, because they run all these samples, and then you have to keep the data, and you need to have all the IT stuff set up to be able to support and store all that data. So it was definitely something. If you're a lab wanting to start doing sequencing, that's a component that you do have to think about. So, yeah, it's another one of the challenges and fun things about sequencing, but it's worth it in the end. So, well, good. So let me think. When would you like to meet next to kind of go over stuff again?
I think from tomorrow, like any time between after 2 or like that, will it work, or after 1:30? Because since Dr. Nara is not this week, what I'm planning is I know because first I was in the clinic, so I know many people. So since Dr. Nara is not here, and I can cover this stuff as well. It's not necessarily for me to be in the lab for this stuff, what I'm doing right now.
No, absolutely. Yeah, you can do it from anywhere.
So I wanted to go to clinic to see some more patients.
Perfect.
I want to do this week only. Once Dr. Nara comes, I will be coming to the lab as usual.
Okay, that's fine. Okay, so then do you want to do, so you said after, do you want to do 2 o'clock on Tuesday? Would that work for you?
Yes.
You'll be done with clinic by then?
Yes, mostly I will be done by clinic at that time, and I can go to lab or I can come home. Anything is fine.
Okay, so let's plan for 2 o'clock tomorrow, and then so we can do that. I think Wednesday we could meet. Thursday I'm pretty busy.
Okay, no problem.
Especially in the morning, so we may not meet Thursday.
Okay.
But and then Friday.
Yep.
I have some time as well. So, I mean, you're cruising along and doing great. So as much as, I wouldn't feel the need to hurry through this stuff, for this week.
Hurry. I'm also very slow. I'm not hurry. If I would have been hurry, I would have done it.
No, you're good. And so, okay. So I'll send the link out for tomorrow's 2 o'clock, and then we'll talk to you then.
Okay.
Okay. Have a good one.
You too.
Bye.
Bye.